I don't know. That's my question. How important?
Okay, hey you guys on Zoom. We have a question. So I just got on Zoom with everybody else. So we have a question for everybody who's on Zoom. Let's see if we can pull up the chat. So Dr. Young wants to know. I didn't start class yet. Wants to know if y'all would be interested in a prep class for the MCAT. So it would be an online at a $1,000 discount for a total of $1,500. Yeah. Go ahead.
Sure. Hey. So we're trying to. I've talked to Tristan about this, but we're trying to get a course that is an MCAT course that it's an online course that helps you to prepare for the MCAT. I'm just wondering if that's something that y'all would be interested in. Do you think that it's useful? Like some students say that it's not useful, that it's not really worth the $1,500 that you would spend. And then others say that it is useful to have an online course. And so I sort of want to know what is the general consensus among students. Can they just share in chat?
Yeah, they can share in chat.
Just share in your chat box, okay?
And I put the volume up.
Can we get a joke? Seriously?
Hey, my jokes aren't good enough for y'all?
No, it's not personally it's you. It's that Dr. Young also has pretty spicy jokes.
Spicy.
Mine are not spicy.
Any ideas about the MCAT course?
No, it's just
The MCAT course?
It's called.
Or, look, email me if you have an opinion. If it's something that you would like to do, an MCAT course at Nichols, or not at Nichols, excuse me, it would be an online course, and it'd be about $1,500. I really want to know, do you think that it would be useful?
Would there be like a professor teaching tips?
They have their own professors. So Courtney asked, would there be a professor teaching with like tips for taking the MCAT? I assume so. In Kaplan, they have different instructors that teach the different content areas. Okay, well, I went to get blood the other day, and you know, they type your blood, like they give you your blood type, and they told me that I had type A blood. I said, that's not right. It's a type O. All right, email me, okay, if you have any ideas.
He's got a lot of practice.
I'll let you know what to think, okay? All right.
All right. So don't forget to email him if you have an opinion either way, whether it's no, I would not be interested at all, or yes, I would be. But he's a dad. He has better dad jokes. I'm a mom. Mom jokes are no good. I get no love.
Dad and mom jokes. Dad and mom jokes are. Dad and mom jokes.
Okay, Muhammad, tell us a joke, please. Please tell us a joke. Save me today from having to tell a joke. Muhammad's going to take over today. If y'all need a handout for the chapter 6 long questions, you can grab one.
Thank you.
All right, and I need to start. I haven't started the cloud. What do you do with a chemist? I don't know. What do you do with a chemist? Bury him? Bury him?
Bury him.
Bury him?
You bury him. Not bury him. You bury him.
That was good. It was good. It was very good. I like that. I know I put a joke in my son's lunch today, but I can't remember what it was. I don't know.
Yeah, Dr. Young has great jokes.
He has great jokes. I just have the third. Well, it's really hard for me to find jokes that Charles will understand.
Yeah.
If he'll get the, oh, how does a flea move from dog to dog? Through itch hiking. So Dr. Young gets a round of laughs, and I get, oh.
Maybe
it's delivery.
For more things.
Yeah. Well, as Charles gets older, my classes will get happier.
Somehow like you learn Corey jokes the way you got dad, because apparently my The same thing with my dad.
He knows some good corny jokes.
Yes, he does.
All right, let's see.
Welcome to
Why-
the sixth grade.
Why do you study for chemistry? I don't know. Why do you study for chemistry?
Because it won't have to.
Other than you don't want to fail. That's a good, legitimate. Because chemists try. Because chemists try. That's. I like that one. I really like that one.
Yeah, we're a comedian.
I really like that. I'm gonna have to use that. Okay, let's see. So I think I have everything going. Oh, I need to do attendance. I need to do attendance. Look, I'm laughing so hard I'm crying a little bit, you know? I know that's bad about my sense of humor. I'm usually the only one laughing at my jokes, so. Right, exactly.
Yeah.
I crack myself up, and you know,
That's
that's
on your face.
matters.
So self.
All right, here we go. So go ahead and do attendance, whatever you can do attendance. And let me minimize that. Come back to this. Full screen. Okay. Oh, and I'm gonna try one more time. It's really nice when I have the chat on my laptop. It doesn't always work, but it's nice. So I'm gonna try and do that. Section 5. Oh, and I have extra section 5 printouts if you wanted them. You know, I know some people
All right, well,
didn't. They're up here. They're good to go. Okay. So here we go. This is what we're doing today. Okay. For right now, I'm just gonna have to leave the chat up on this screen. All right. So you guys kind of had a chance to look at these long eye clicker questions, right?
Yeah, I have.
Yeah, and you've thought about them, got some ideas?
Can't guarantee I understand them or you should know them.
Well, we're gonna try and understand them better. That's our goal, right?
Exactly.
Okay, so in the first one, in number 1, we're comparing the amino acid sequence for 3 different globin genes. Human myoglobin, beta globin, alpha globin, right? So what's boxed in?
Yeah, and
Why do we box these helices in? Like, why is that helpful? Because all of them have 8 different helices. That's the general overall structure of a globin, right? And it's these subunits that get put together, right? Okay, good. So now the next question is, what's important within those helices, right? So let's see if I can move this down. All right, so everybody had a chance to check in on attendance before I close it? Anybody need a second? I have 1 more, that's good. 2. All right, 3 minutes, I'm gonna close attendance. So if you have not done attendance, please, please, please do attendance. Okay, all right, so I want you to click in what your answer is before we start analyzing. Why did I do that? This one. So click in your answer to this one before we discuss, and then we'll discuss and compare our results. So while you're clicking in, y'all can answer this. Why did we highlight here E7 and F8? Like, what is E7 and what is F8? F8 is the proximal histidine. What does the proximal histidine do? Get away, don't get away. What does the proximal histidine do? It's below the what? below the heme group. So you have basically these two residues that come in from the top and bottom to help to stabilize the heme, and the one at the bottom stabilizes the heme, the one at the top is what binds to the O2. So then we get a complete stable, and that's our oxygenated form, right? Okay, so the proximal His is down below. Why do we call it? Y'all know proximal and distal? Y'all know those terms from biology? Proximal meaning closer to the point of attachment, distal meaning farther away. So the His is closer to the heme group on the bottom than the one on the top, so they call it the proximal. Okay, so how about E7? What is E7? If that one's proximal, what's E7? Distal. Why do we call it E if it's histidine? Because it's in the E helix. It's the 7th residue in the E helix. Okay. F8 is the 8th residue in the F helix. All right. So A says histidines at positions E7 and F8 are conserved in all 3 proteins. Is that part of the statement true? Yes. How do you know that? So this part is true. Look, we have an asterisk, HHH. We have an asterisk, HHH. They're all conserved. Now, you have to evaluate the rest of the statement. Because they are amino acids required to coordinate the oxygen binding in the heme cofactor of the given globulin molecules. Is that what they do? Yes. Yes, that's what they do. They coordinate oxygen binding and heme. That's what y'all just told me it does, right? Yeah, I can be a little bit tricky. Oh, but I'm supposed to stop this. I'm not supposed to let y'all answer after we've evaluated our statements. All right. So A is true. 1 or more. Yeah, 1 or more. Okay. But any answer choice that you pick that is correct will show up as correct. All right, B, mutating the threonine just to the right of the F8 amino acid. So let's go up the F8, and we're going just to the right. So which residues are those? ACA. Okay, that's the ones we're talking about right here. So mutating the threonine just to the right of F8 amino acid in human myoglobin to an aspartic acid would almost certainly reduce or eliminate correct functionality of the protein. What could you tell me about that? There is no threonine. There is no threonine. That's number 1. And is it highly conserved? Not highly. I mean, it's not all over the place, but it's not super high, right? So, and cysteine is pretty different than alanine, right? And everything's still functional. So you could probably have a lot of variation there and not have a big deal. Okay, so we're going to say no on B. Mutating the glycine immediately to the right of E7 shown in human myoglobin to a tyrosine. So let's see, we're going to go to E7 and we're going to go immediately to the right.
But it's all glycine.
It's all glycine. Okay, so mutating the glycine to a tyrosine would likely reduce or eliminate the correct function of the myoglobin. Why? It's highly conserved. It's highly, highly conserved. And what are you doing when you convert from a glycine to a tyrosine? We go from, what is glycine? Yeah, but like, it is the smallest residue. It is the smallest residue. And then you're going to stick a huge honking aromatic in there. Something's going to go wrong. Right? That's what you kind of need to be thinking about. So I would say this one is good. You're changing size. That's right, Hannah. You're changing a lot of size. And if you have a glycine that's highly conserved, very close to a residue that you know interacts with another protein or with another functional group or something like that, then that's highly likely that that functional group that's participating in the interaction needs the space. And so glycine's kind of hanging out down low so that this other residue, this other side chain has space to do its job. Otherwise, you put in another one and it's going to have a steric clash and it's going to move it. Okay. It is equally reasonable to assume that changing amino acids at either conservative or non-conservative substitution sites will yield similar results in terms of the structure and function of a globin gene protein. Only at conservative, only at conservative. So you would want to get rid of that one, right? So that one would be incorrect. How we feel about that? Pretty good. So let's see what we said. So could have been A or C. A or C. Look, y'all are doing good. Doing good. All right. Okay. Questions on this one before we move on to the next one? Okay. All right. Now, I'm just going to put the answer choices there so that you can see the whole thing on here. So I'm going to give you a minute to go ahead and put in your answers, and then we're going to talk about it. So what did you put for number 2? Give y'all a minute and a half to get all your answers in. Oh, and everybody got the notification that exam moved to Monday? Yeah.
Hi guys.
Okay, good. And I need to make that phone call. I'll do that.
Okay.
I had meetings this morning, so I'll look into it.
I need to talk to myself if need be.
Okay. All right, so a minute and a half, but not everybody has checked in. We're supposed to have like 50 something. Oh, you did, Hannah, you didn't know? Hannah moved to, you got the announcements. Yeah, Hannah, you can talk to me after class. Yes. Okay, come on, y'all. How come we only got 47? Was it more than last time? Y'all. Okay. All right, so hemoglobin Syracuse is a human hemoglobin with this mutation, His 143 to proline 143. So tell me, before you keep reading all of this information, tell me about those residue changes. Histidine is positive. What is proline? Non-polar. And sometimes it's important when you're looking at these mutations to not only say, okay, what classification was it in, but compare it to what it was originally. So if it originally was a His 143, His is positively charged. So tell me about the charge on our new proline. Is it positive? Give you a hint. No. It would be neutral, right? So neutral, and then you can put non-polar behind it, you know, but sometimes that helps you more than just saying, okay, what classification was it in the book, you know? Okay, so we have this mutation. Now we're given another piece of information. In normal adult hemoglobin, His 143 interacts with 2,3-BPG. Ooh, so this is a residue that actively participates in an interaction with another molecule. Tell me about 2,3-BPG. Okay, so the interaction site, the side chain where BPG interacts with hemoglobin is a negative side chain. You do have a negative side chain, but you weren't given that information, and you might not know that. What do you know about BPG? What does it do? What's its role? It's a negative allosteric regulator of what process? O2 binding to heme, right? So once 2,3-BPG sits, it sits like right in the middle of the molecule, it prevents all 4 of the subunits from binding oxygen because it creates a conformational change in all of those subunits to decrease the ability to bind oxygen. Okay. So then we're told, and I love to do this on these questions, just kind of jot down what you know. The oxygen binding curve for normal adult hemoglobin under physiologic conditions is given, and in the presence of 2,3-BPG. So we're telling you that there's a negative regulator there. How would the oxygen binding curve for hemoglobin Syracuse under physiological conditions differ from the normal curve? So how did you draw your curve? Shift it to the left. Why did you shift it to the left? Because what are we looking at here? This is fractional saturation versus our concentration of O2 in free O2. So if we make this mutation, we go from his to proline, what's going to happen to binding of 2,3-BPG to our mutant protein? So if you have mutant plus 2,3-BPG, what's going to happen? BPG won't be able to inhibit. Won't be able to inhibit O2 binding. All right? So if 2,3-BPG can't inhibit, it's just like taking out 2,3-BPG from our reaction, right? So what's going to happen to our fractional saturation? What is fractional saturation, by the way? Yep, fraction of occupied binding sites. So if we take away the ability to interact with 2,3-BPG, then our curve, and I'll do it in red, our curve should be shifted and it should be more hyperbolic. Okay, so let's look at our answer choices, right? We did all that work and we didn't even answer it yet. All right, so A says there would be no change in the binding curve. Well, automatically, what are you going to do? No change, it's not going to happen. They're saying this is a silent mutation. Is this a silent mutation? A silent mutation would do what?
Nothing.
Nothing. You would have the same residue there. So A cannot be it. Am I still going on? Yeah, okay. Good. No change in the sequence, right? Okay, B, the mutant binding curve should be shifted to the right of the normal hemoglobin curve since the mutation increases hemoglobin's affinity for 2,3-BPG. Does it increase affinity? No, it decreases. False, it decreases, good. Resulting in a higher propensity for the T state. That is not right. And what's the difference between T state? You do need to know T state and R state.
He said deoxygenated and R state is oxygenated.
You want to know how I remember this? If I don't have oxygen, trust me, I am stressed. I am tense, right? If I have oxygen, oh, I can relax. Oh, I can take a big, deep breath, right? So that's how you can kind of remember that. And look, if you need to make yourself a little story to remember some things, do it. No big deal. All right, C, the mutant binding curve should be shifted to the right of the normal hemoglobin curve, as the mutation will make the 2,3-BPG binding less stable. Well, that's true. It will make binding less stable, so the affinity for oxygen will, will what? Is the right. I'm like, that was right. So we want to go to the left. So it will become less stable and it will increase. So this part is correct and this part is correct.
Represents the oxygen that is free, so when oxygen increases, there's less free oxygen.
Yep, down here, this is free oxygen. And here, this is binding sites that are bound to oxygen. Yep. Okay. All right, D. So we had to X out C, we had to X out B. The mutant binding curve should be hyperbolic and to the left, ooh, now we're getting somewhere, of the normal hemoglobin curve, as the mutation will make 2,3-BPG binding less stable. So the affinity for oxygen will increase. So that's right. There we go. All of that is correct. So what's wrong with E?
It's saying that it increases hemoglobin's affinity.
So this is saying that it increases hemoglobins, and that should be opposite. It should decrease. Good. And it should result in a higher propensity for the R state. So how did we do? Where's my, there it is. Oh, we were kind of all over the place. Does that make more sense to y'all now that we've kind of discussed it? Okay. All right. All right, so you watched your animation for hemoglobin, right? They have some really good animations. I really like their animations. I think they did a good job. Okay, so put in your answer and then we're going to talk about it. Y'all should already have answers for these before you walk in the door, right? So I'm calling it a minute 30, so everybody gets a chance to put their answers in. I think 48 was our max. I'll wait the full, it's just 10 seconds. Okay, so All right, high concentrations of 2,3-BPG and carbon monoxide both inhibit oxygen carrying capacity, right? And we talked in detail about 2,3-BPG, right? And 2,3-BPG binds at that interface of the two beta subunits and stabilizes the T state. So it promotes the conformation of the T state. Carbon monoxide, on the other hand, binds to the heme iron in the same manner as oxygen. So what do you expect the carbon monoxide binding shift to shift the equilibrium in favor of the T state or the R state or neither state? So what did y'all say? Ooh, we're split down the middle. We're split down the middle. All right. We know that when 2,3-BPG, right? When it binds, what you're gonna have are side chain interactions between the hemoglobin and the 2,3-BPG, right? So this induces a conformational change. And that conformational change stabilizes what state? T state. Okay, tell me about O2 binding. It's the opposite. Do we still have a conformational change? Yes, we have a conformational change, but it's the conformational change, C-H-A-N-G, change to stabilize the R state. How does O2 binding stabilize the R state? So when the oxygen interacts with that iron, right? It makes an interaction with those electrons of the iron. How does that help to stabilize the R state?
Because it causes because when it comes in contact with the iron, it causes the iron to like contort or twist into the other.
Well, you got to think that this iron atom takes up so much space, right? What takes up space in an atom?
That's great.
What determines its radius? Electrons. Electrons. If this iron atom participates in an interaction with another atom, like an oxygen molecule, what's going to happen with those electrons? They're going to go and be shared with our other atom, right? So what happened? So the puckering is when iron is big, it puckers. When O2 binds, the diameter, the radius of the atom will decrease and it'll stabilize, and we don't have that puckering anymore, right? So that's going to stabilize the R state. Okay, now what does that have to do with CO2? How does CO2 interact with hemoglobin? What does it say?
That's carbon monoxide.
That's what I meant, carbon monoxide, I'm sorry.
CO1.
Yes, carbon monoxide, I apologize. So what does it do?
Doesn't it take up the space? Wait, the carbon monoxide poisons because it binds to the iron instead of the O2, which makes where you can't process oxygen.
Okay, so it binds, so the carbon monoxide binds to our heme in the same way that O2 does. That's exactly right. So now, if it binds in the same way, tell me conformationally what's going to happen. The same result as O2. So what's going to happen? If we have carbon monoxide, we're going to get a conformational change. What's it going to do? It's going to favor the R state. So the heme will be in the R state, but will it have oxygen bound to it? It'll be in the R state with what? CO, carbon monoxide. This is why carbon monoxide poisoning is so bad, because just like Ross was saying, right? We're taking up the oxygen binding sites. So hemoglobin thinks it's carrying oxygen. Is it? No. Nope. It's carrying carbon monoxide, and we can't do anything with that. So you will starve your cells and you will die. Right? Okay, so.
So it's like you can breathe, but you can't. It's like your
body thinks it's able to breathe, but in actuality it
In
can't.
actuality, it can't. So
That
what's,
sounds like a very torturous way to die.
well, you don't really, you just get tired. Just get tired. And you go to sleep and then you don't wake up. Okay, so, so what answer choice should we have said? The R state is favored. The R state is favored. So that's the oxyhemoglobin state, but it's bound to carbon monoxide, right? Okay, so Allie says, are the test questions going to be similar to these type of longer questions? You will have 2 discussion questions on this exam, and it's going to be a combination. It's not. You will have things where you have to think, right? And you'll have some that are more recall. So you'll have a mix.
So it's gonna be kind of like a mixture like this and like our clinical questions.
Yep.
What
Yep.
about the discussion questions?
Similar, yeah. So those are a good place to go to like get more practice with discussion questions. Absolutely, absolutely. So the question was, the thing was, is it good to look at the discussion questions at the end of the chapter? And the answer is yes. You'll have less questions, so there'll be half as many discussion questions on this exam as there were on the last exam. So you should have a little bit more time to think and construct answers.
What about the self-check?
The self-check's great. Self-check's great. I try to give you all resources that will help you. And I try also to make, like, I know that these long questions are difficult. I know that. But I try to make these long questions more difficult than what you're going to see on the exam. Because if you can handle this, you can handle the exam. I don't want you to think we're just going to do easy questions in class and then it's going to be hard on the exam. I don't want to do that to you.
So
Right.
basically, you're trying to do the opposite
Mhm.
in a sense.
It's like conditioning for the exam. Yep. Yep. If you got this, I can handle the exam, right? Okay. Here we go. All right. So, do we need a calculator?
Yeah,
I think you do.
There's one thing,
Yeah. Yeah. You should have a calculator. Yes. You should have a calculator. Yes. Okay. So, ooh. So you watch the animation on muscle contraction. Y'all know that this is like one of my favorite things, right? Actin and myosin. I skipped 4. Oh, because it's at the bottom. I was just going down. Thank y'all. Thank y'all. Okay, we skipped 4. Thank y'all. Y'all got to keep me on track. All right. So if identical samples of human blood are treated with different concentrations of chloride ions, the samples with the higher chloride ions we found to have a higher concentration of free oxygen in solution, that is, not bound hemoglobin. Based on this information, would you expect chloride ions to stabilize the T state or the R state, and have an answer as to why? So go ahead and click in your answer, but do know that you have to explain why. You don't need to know the binding affinity. You don't. I'll talk about why. I'll explain it in a minute, Tristan. Okay, a minute 30, I'm calling it. All right. So I want to see kind of what people said. All right. Okay, so what kind of information were we given in the question?
It does not find
Okay, we were told that, right, the higher the chloride ion concentration, right? So if we increase chloride ion, what happens? Increase in free O2. Okay, we were given that. Were we given anything else? No. But we have to be able to relate free O2 to the T state versus the R state. So can we do that? Do we have enough information to do that? Yes. So tell me what happens if I have a lot of O2, free O2. What state do you think I'm in?
It means unable to bind to the T state.
If I have a lot, then I'm going to be in the T state, right? I'm going to be in the deoxy state. That's right. So the presence of chlorine does what? Because if we, the sample saying that the higher Cl, higher concentration of free also says, infers low Cl, low free O2, right? So that would be our state. So what can you say? Well, in the presence of Cl, so as we increase Cl, what are we going to do? Stabilize that T state. Okay. So did you have to have a binding affinity, Tristan? This was one of those more logical things. If you can infer what does a lot of free O2 mean, then you can answer this.
So free O2 means it's unable to, if there's more free O2 in the sense, that means it's unable to, means that's more, it's unable to bind to the hemoglobin, which means more hemoglobin in the T state.
Correct. Because your oxygen can either be free oxygen or your oxygen can be bound oxygen. Those are your only two options. Right? Okay, I didn't mean to act as a competing ligand with free. I mean, I didn't know that chlorine. What? Didn't know that it doesn't act as a competing ligand. Could it act as a competing ligand? No, it acts more like 2,3-BPG. Tristan, this is, it's negative. So it's a negative, yes. It's a negative, yes. Yes, yes. Okay, are y'all ready to go on?
Yep.
So do we have to know? No, you don't have to know about chlorine specifically. You have to be able to pull out conclusions from information given, knowing that hemoglobin can be positively and negatively allosterically regulated. Does that statement make sense to y'all? Yeah? Okay. All right. So now, my favorite. Y'all know my favorite protein on the planet is active. It's my favorite. Yes. Okay, so you watch your animation on muscle contraction, right? So you have 1, 2, 3, 4 answer choices. So just put them in, short answer, and then no spaces, right? The word, comma, the word, comma, right? No spaces. So we try and get them as uniform as possible. So go ahead and put in your answers, no spaces. And I know this is going to take you a minute to type, so I'll give you a little extra time. I was hoping for about 2 minutes. We have 19 responses. So a minute and a half, 2.5 minutes, excuse me. So another 35 seconds. Is it more trouble to put in? Is it a lot of trouble to put in? Just takes a minute.
Is the third test going to be still where it's at right now? Or is that going to move back?
We'll move back one. I think I already moved it back. I think I already updated it on the calendar. So it's, I think, on drop day now. I think.
It is, I was checking.
Yeah.
So the drop day is on the same day as the third test?
Correct.
Okay.
Correct. All right. 3 minutes and 30 seconds. I'm calling it.
Will we have the sign up sheet to do the test in class?
Oh yeah, y'all want to? Yeah. I mean, I don't think so few people showed up to do it in class. I'll do it.
I just say that because I have to come to school anyway, so it's either going to be in the library
Or in here. Yeah. So I'll put that out and I'll send an email to everybody that says, hey, the sign up is up, and I'll put it on Moodle, like where I put the exam. I'll put the sign up right there next to the exam. Okay. So yeah, I will do that. Anything else y'all need for that? I think that should be it. Okay. But I will put the sign up sheet up. Okay. Only 30 responses. My numbers keep going down. This is so sad. All right. So in the sliding filament model of muscle contraction, the blank filaments are made of myosin. Thick filaments. Thick filaments. They call them the thick filaments because myosin is made up of a long, skinny part and then a head domain, right? And so all of these long, skinny pieces line up, and then they have heads sticking out. And so what ends up happening is that you get a really, really thick part of all the myosin bundled together. And so that would be why they call it thick, because some of the filaments look thin, some of the filaments look thick, the myosin looks thick. Okay, how about, and the blank filaments are made of actin. The thin filaments are made of actin. And they're what the myosin basically grab onto, right? And it's a kind of a helical, double-stranded polymer. They're both polymers. Okay. All right, so thick and thin. All right. The two filaments slide past each other during muscle contraction, shortening the distance between, what could you have said? Z-disc. Z-discs. Right? Because that's how a sarcomere is lined up, right? And that's why they call it a Z-disc, because it looks like a zigzag. And then you have what's bound to the Z-disc. Hint, hint, hint. I'm drawing it. The thin filaments are hooked to the Z-disc. And then what do you have going, starting from the middle and then kind of moving outward? Right, that binds to the actin, and it goes this way too. Right, and then you have another layer on top of the actin. And I'm not the world's best artist. I apologize.
Don't worry, I know exactly how you feel. In fact, I actually got a C on one of my old test because I couldn't draw. I couldn't draw like this the sperm cycle,
Yeah.
couldn't draw a sperm cycle properly.
It happens. All right, so we've got thick and thin filaments, we've got Z-disc. So what makes the contraction happen? The binding of calcium, Ca2+, right, to troponin. And troponin is a little cluster of proteins that sits every so often on the actin molecule. And so when troponin binds to that calcium, it induces a conformational change in yet another protein that sits along the actin, and we call that tropomyosin. So when tropomyosin moves out of the way, myosin can bind to the actin and pull it. And so you got to think about, this is how I equated the analogy. You'll remember this, right? Let's pretend I have ropes that are attached to the sides of the classroom, right? Those ropes are going to be actin. If I grab those ropes with each hand, and y'all at home, I'm sorry. If you grab those ropes with each hand and you pull them in, that's the myosin pulling the actin. And so if I were strong enough, right, I'd be able to pull these walls in closer together. So from wall to wall is like a sarcomere. Well, there's another classroom right next door. Let's say the teacher in that classroom did the same thing. They grabbed the ropes that were attached to the wall and pulled them in. We wouldn't be able to pull very far, but if you add up all the changes from all the classrooms, you would have the muscle contraction of your entire arm, right? So what you're doing is a small change in the sarcomere, but you have tons of sarcomeres. So this allows you to see a big movement, okay? All right. So now you do have to know the actin-myosin cycle. So it's a 5-step process. Oh, I did not. Thank you. Thick, thin, Z, calcium. Okay, thick, thin, Z, calcium. That'll work. That's fine. That's fine. Thick, thin. No. Thick, thin, Z, calcium. Thick, thin. Whoa. Thick, thin, Z-disc calcium. Z-disc calcium. Thick, thin, Z-disc calcium. Thick, thin, Z plate. And thick filament. Thick, thin, Z-disc, yeah, calcium. Thick, thin. Tell me if I missed one. Thick, thin. Okay, I think I got them all. All right, let me know if I didn't. Let me know if I missed one. All right, so now you have to know the whole process of how that myosin will actually bind and pull on the actin, right? Because it's like those head groups reach out, attach, and then crank them in. So it's like a ratchet. All right, so you're gonna put them in order. So when you answer, in short answer, just do capital letter A, comma, capital letter B, comma, C, no spaces. No spaces, okay. I guess this will be our last one today. We got one more, but we got Friday, so it's okay. And I have a few more clicker questions too that we never got to, so we'll be able to catch up on those. Which clicker questions?
Like also, the original ones, like the short ones where you
That was to ease you into things, Ross.
Okay.
You have a few easy ones here and there. So I think using the animations and going back and trying to see how things work is a really good study tool for like this and for hemoglobin binding, things like that. Those animations are really awesome. They're going to be long clicker. Questions to print? No. Hannah, there'll be one last long iClicker that we didn't finish that's already on the same packet, so number 7. And then there'll be short clicker, like that I'm doing the PowerPoint slide sort of deal. Mhm. All right, I'll give you until 2 minutes, so get those answers in. Anybody left? We had 38 before. We keep dropping like flies. Oh, 37. One more. All right. So what did we say? So what happens when we add calcium? What's the response when calcium comes in? What's it going to do? Bind to troponin, right? Troponin. So that's C.
Troponin, troponin. Honestly, I feel like this is all words that could be pronounced a multitude of words.
There are, and I think the person in the video pronounces it differently too, but it's troponin. I did actin research. It's troponin. All right, the power stroke. What happens in the power stroke? Remember, this is where the power from your muscles come from. What is the power stroke? It's D. Our actin filament is pulled towards the myosin tail. Where's the myosin tail? This is in the middle of the sarcomere, right? All right, then ADP release. How about ADP release? This one, B, ADP release leaves an empty nucleotide binding site. So it's important that the myosin lets go of that ADP so that we could get ready and primed for the next power stroke. Okay, so ADP leaves, ATP comes in. When ATP binds, what happens? The myosin lets go. Right, it's almost kind of backwards, right? You would think ATP binding, now I'm gonna have the power stroke, but it's not. It's actually the release trigger. Okay, so that's E, so that's the release trigger. And then it releases. What happens when ATP hydrolyzes? So what does that mean? It returns to the conformation. If I grab the actin, I ratchet it, then I let go, what's my return action look like? Right? It returns back. So it's the opposite of the power stroke, right? Okay, so that is A, right? Okay, so let me grade them. So if you have any questions, bring them to class on Friday. And before Friday, I will post the sign up to sit in the classroom. Okay? Y'all want to sign up at the beginning of class? Y'all want to do that in the beginning of class so everybody has an equal chance? Okay, so that's what I'll do. I'll put it in and then I'll open it to become available when we start class on Friday. So CDBEA. Okay. Come on. Okay.
Dr. B,
Yes, sir.
I still don't understand question 4. What I was trying to ask is that
Oh, I'm sorry.
when you have carbon monoxide, carbon monoxide binding to hemoglobin also leads to an increased amount of free oxygen in the blood, right?
You're talking about number 3? Or
4.
number
4.
Okay, and number 4, oh, yes, it does. Carbon monoxide, yes.
So carbon monoxide also induces the R state, correct?
Carbon monoxide induces the R state. Chlorine induces the T state.
Yes, but
Both lead to an increase in free O2.
yeah. So given the information that it leads to a increased amount of O2, free O2, either the T state or the R state could be utilized for both of those.
There are very few molecules that are going to act like oxygen, so you would be given that extra information.
Right, but see, I don't know how chlorine acts with oxygen, so I can't determine that. You see where I'm coming from?
I do see where you're coming from. I do see where you're coming from. I do see that. I do see where you're coming from on that. Yeah. I'd have to give you an extra piece of information.
Yeah, yeah.
Like chlorine interacts, like chlorine does not have a binding site similar to O2. Like if I gave you that in number 4, if I also said Cl- does not, right? Like I did up here in number 5, I said it binds to heme iron in the same manner as oxygen. So I would say the opposite, does not bind like.
Well, to the iron molecule, not to oxygen, right? To the ferrous ion.
Yes, yes. The carbon monoxide binds to the ferrous. It binds to the iron. But what I'm saying here is that the Cl- does not bind in a similar fashion as O2.
Yeah. That's what I meant when I was asking about affinity, because if it had a strong enough affinity, then it would be in the R state, and there would still be a increased amount of free oxygen in the blood. It would be in the R state, though.
Yes, I see what you're saying there. I would have to give you a little extra piece of information. I was assuming that since you didn't have the information, you can't say that it binds. So then you can't say it's R state.
Okay.
But no, I understand what you're saying, and you would need that piece of information. So when I was writing this, I didn't think about that in that way.
Okay.
But you're legit. You're good.
Okay. And the first question, you I don't want to take up too much time, but you said top and bottom proximal and distal histidines, but those are relative terms, right? That's not going to be on a test of like bottom one is always the proximal one and top one's always the distal one. It's just a relative thing.
What do you mean relative?
Like there's no top and bottom. You can orient the molecule however you want.
Oh, right. That's correct. That's correct. That is correct. There is no top and bottom. Proximal and distal are telling you distance from the heme group to the amino acid backbone.
Mm-hmm.
So the proximal, the heme group is closer to the backbone. The distal, the heme group is farther away from the backbone.
Mm-hmm. In the textbook, it describes heme as a prosthetic group. Is that the same as a cofactor?
Yeah, I'm not gonna call it that.
Okay.
Call it that.
Also, part D of question 1.
Part D. Okay.
Yeah. Answer choice D.
This one?
Mm-hmm.
Okay.
It's a conservative that's okay to make changes in, or more okay to make changes in?
No. You cannot.
That's what I thought.
You cannot make changes at conservative sites.
Yeah. Okay.
Without inducing a change, right?
Yeah. Yes.
Yes.
And final question, number 5. I put between Z disc and the thick filament. Don't the Z disc and the thick filament get closer together during the contraction?
No, they do, but that is not how we determine the contraction. You measure from Z disc to Z disc. That's the sarcomere. It's all about the length of the entire sarcomere. If you did that, then where are you measuring? Like what part of this are you measuring to the Z disc?
Isn't there a part of it called the I what's the different parts of it? The I fragment and
the I band, the A band.
Yeah. So the I band's getting shorter, so doesn't that mean the thick filament's getting closer to a Z disc?
Yes. But that is not what we measure. What we measure is the distance from Z disc to Z disc.
Okay.
So yes, the I band gets smaller. The